Thank you for introduction. I'm Daisuke Oida from University of Tsukuba. Today I'm going to talk about computational multi-directional optical coherence tomography for visualizing the microstructural directionality of the tissue. The target of our research is quantification of microstructural directionality. The assessment of microstructural directionality is beneficial in some clinical fields. For example, myocardial fiber structure is related to the heart functions. The organization of Henry's fiber layer, which is a layer of retina, is useful for diagnosis of retinal diseases. And the collagen fiber in connective tissue, such as tendon and ligament, is associated with the biomechanical property. To examine these microstructures, some approaches has been taken. Histological assessment is a basic method for evaluating microstructure, but is invasive and time-consuming. Diffusion tensor magnetic resonance imaging can visualize three-dimensional fiber structure of the heart, as the resolution is limited to sub-millimeter. And SHG microscopy can observe collagen in the tissue, but the imaging depth is limited to several hundred microns. To overcome these drawbacks, we focus on the technique of optical coherence tomography. Optical coherence tomography is an imaging modality which measures three-dimensional morphology of biological sample. And OCT provide non-invasive, high-resolution and high-penetration imaging. But conventional OCT doesn't have enough resolution for tissue microstructure. So then, some extended functional OCT methods were developed. For example, polarization-sensitive OCT tractography can visualize nerve fiber of the brain and the myocardial fiber by using the polarization property of the tissue. But the hardware system is complicated. So in this research, we focus on the technique of multi-directional imaging. In this technique, sample is measured from multiple directions. Here, if the sample has microstructural directionality, light intensity reflected from each direction are slightly different. From this difference, we can subjectively guess a microstructure. But it is utilized only for observation, so it cannot be utilized for evaluating microstructure due to low quantitativeness. Then how to improve? There are two ideas. One is increasing the measurement directions. Some groups revealed Henry's fiber layer by measuring from several directions, but it will cause long measurement time. Another idea is increasing the number of beams. The group in the Medical University of Vienna measured samples from multiple directions simultaneously by using multiple beam system, but the measurement direction is limited due to hardware complexity. To solve this problem, we developed computational multi-directional OCT. In this method, multi-directional imaging is realized by signal processing of complex OCT signal. Since the signal processing is performed after data acquisition, multiple directional imaging is acquired without increasing measurement type and hardware complexity. Signal processing of complex OCT signal is achieved by a sequence of two types of signal processing. At first, OCT intensity analysis is performed for determining the dominant emphasis fiber orientation, and then phase analysis is performed for visualizing the depth-oriented microstructure directionality. So then, how to implement this technique? Let me start with system configuration. In this study, we used a standard spectral domain OCT system with no hardware extension. The theoretical, actual, and lateral resolution of this system are 3.1 and 4.9 microns, and the measurement speed is 50,000 airlines per second. From this system, we can obtain the volumetric OCT data. And then OCT intensity analysis is performed. Before OCT intensity analysis, bulk phase error due to the system imperfection and air turbulence was numerically collected. And then one emphasis OCT intensity slice was extracted, and it is Fourier transformed and binary numerical mask is applied. And then inversely Fourier transform to obtain intensity directional images. Regarding the mask, shape is a pair of one shape which locates symmetrically in terms of region. The central angle is 20 degrees, and the radius is 5 to 50 cycles per millimeter. To obtain dominant emphasis fiber orientation, we generated intensity directional images while rotating the mask from 0 to 179 degrees. each rotation, total pixel intensity is computed and plotted as a function of rotation angle. And we can obtain the plot. At first, calculate the center of gravity around the peak with a range of plus minus 15 degrees. And then dominant fiber orientation is determined by 90-degree difference from the center of gravity. And then phase analysis is performed. In phase analysis, first, one emphasis complex OCT signal is extracted, and then generate phase-only signal where the amplitude is unity. And it is Fourier transformed and numerical mask is applied and inversely Fourier transformed to obtain phase-directional images. For multiple directional imaging, several types of numerical masks were prepared. The shape is a pair of one shape and the central angle is 180 degrees. The radius is 110 to 210 cycles per millimeter. Regarding the direction, here if the sample has such a dominant emphasis fiber orientation, the mask direction is chosen to be parallel and perpendicular to the dominant emphasis fiber orientation. So the 4 mask is used in total. Then let me show phase directional images of bovine tendon. The insets of each image show the applied mask shape. From these images, we can see some difference, but it is small. So to clearly show the difference, pseudo-color directional contrast images were generated. Let me explain the procedure. First, select 2 phase directional images with 180-degree directional difference. One image is used as red channel and the other image is used as green channel. Blue channel represents the average of 2 images. And phase directional contrast images is generated by merging 3 channels. The color bar of this image is shown as light. The image appears as purple when the red channel is higher than green channel. And image appears as green as the opposite case. And the image appears gray where the intensity of 2 images are almost the same. Then let me show the result and do the discussion. The left graph shows the result of OCT intensity analysis of bovine tendon, and the center 2 images show the emphasis and cross-section OCT images. The left graph has one distinct peak, and the calculated center of gravity was 63 degrees. So the dominant emphasis fiber orientation was 153 degrees, and this direction corresponds to the visible fiber orientation in the emphasis OCT images. The right 4 images show the phase directional contrast images. In the emphasis images, purple and green stripe appeared along the fiber direction. Here the mask is perpendicular to the dominant emphasis fiber orientation. And this stripe also can be found in the cross-sectional images as vertical stripe. On the other hand, homogeneous green color appear in the emphasis images where the mask is parallel direction. So the question here is, what do purple and green color represent? To answer this question, numerical simulation was performed. At first, pseudo-OCT signal was generated based on OCT theory. Here the gamma d is the coherence function of light source, delta d is path length difference between sample and reference arms, and S of dxy is surface shape of fiber. Here the surface shape of fiber was simulated based on a cross-section of OCT. In this image, we can see several sides of cross-section of fiber. And from these fibers, the shape of ellipse fiber was estimated. From this estimation, we generated pseudo-OCT signal which has the ellipse shape of fiber along x direction, and then create phase directional contrast images by using pseudo-OCT signal. At the same time, we consider the sign of delta d because it flips the image along the depth direction. Let me show the simulation result. When the delta d is positive, purple and green stripe appear in the emphasis images. Here the mask is perpendicular to the dominant emphasis fiber orientation. And in the cross-sectional images, purple and green appearance can be seen, and this appearance is consistent with the cross-sectional phase directional contrast images of bovine tendon sample. By zooming out one cross-section, we can see that purple and green color correspond to the positive and negative shield of sample structure. And this relationship also can be seen where the delta D is negative. So, the relationship between the sample shield and color appearance is independent of sign of delta D. And the monochromatic color appears when the mask is parallel direction. Then it shows there is no shield along the fiber direction. By using this finding, phase-directional contrast images can be interpreted. The purple-green stripe shows that the two sides of fiber-rich structure, and green homogeneous color represents that the fiber is shielding along fiber direction. From this interpretation, we can say that phase-directional contrast images visualize depth-oriented microstructure. Let me summarize this talk. In this study, computational multi-directional OCT, which can realize multi-directional imaging by signal processing without increasing acquisition time and hardware complexity, was demonstrated. OCT intensity analysis determined dominant emphasis orientation. In the phase analysis, phase-directional contrast images showed purple-green stripe. A numerical simulation showed purple-green stripe represents rich structure of fiber fracture. The conclusion is our proposed method would be useful for visualizing the microstructure directionality of the tissue. Here, I'd like to thank my shared colleagues and collaborators. Thank you for your attention.